All right, so our next speaker this morning will be Dr. Edward Curtis, who is in IOCB, the IOCB Prague, Department of Biology, Prague, Czechia. Let's get set up here, and I'll give my usual comment. Still processing in my mind Czechia versus the Czech Republic, but countries are allowed to name themselves whatever they wish. That's during the Olympics with the hockey, Canada, it always was the Czech Republic, but no. Anyway, I will stop rambling on.
Yep. Well, you can see that I'm a bit old school, sticking with the Czech Republic here, so sorry if anyone doesn't like that. All right, so the kind of traditional way to think about DNA is as a double-stranded molecule that stores genetic information. And while this is undoubtedly very important, it's now appreciated that DNA can form other kinds of structures and also have many other interesting and useful functions. Our group is especially interested in DNA molecules that catalyze reactions. These are called deoxyribozymes. They were discovered relatively recently in 1994. They're usually identified using methods of artificial evolution like cell-X in vitro selection. They can catalyze a wide range of reactions. They can be chemically synthesized, typically in a matter of hours using a DNA synthesizer. They're also very stable and not so expensive to manufacture. So these are some of the reasons our group is interested in catalytic DNA. In a recent study, we identified a deoxyribozyme called Aurora that generates a fluorescent product. Here is the 4-MUP substrate used by Aurora. So it removes this phosphate group, which converts 4-MUP into a fluorescent compound. Here is a time course of the fluorescence producing reaction. Here's a secondary structure model of Aurora determined using a method called comparative sequence analysis. And finally, here's the design of a fluorescent deoxyribozyme sensor that uses Aurora to detect ribonuclease activity. So today I'm going to tell you about our quest to more systematically explore the sequence space of Aurora. So ideally, we would like to know every single sequence that can form a secondary structure and catalyze the reaction of Aurora. We would also like to know the best sequence, the sequence that catalyzes this reaction the fastest. So how might we go about addressing this? Well, one kind of straightforward brute force strategy would be to make a library in which every position in Aurora is randomized. So a nice thing about this library is it would contain every possible variant of Aurora. Another nice thing about this library is it would contain the best variant, but it would also contain, most likely, other catalytic folds that can catalyze this reaction, as well as deoxyribozymes that would catalyze other sorts of reactions. So this would be an incredibly interesting library. However, there are a few problems. So the first problem concerns size. So a library with this design would encode 2 × 10⁻28 possible sequences. So this is a huge number. And if we prepared this library at the standard concentration of 1 micromolar, we would need a volume that's about 10 times larger than this beautiful Czech lake called Černé jezero. So unfortunately, we don't have this amount of space in our lab. But even if we did, there's a second problem, and this involves cost. So the cost of this library would be higher than the total number of euro in circulation in the world. So again, we don't have this money, and in fact, no one in the world does. So today I'm going to tell you about 2 strategies that we recently developed in the group to try to address this problem, this limitation of exploring sequence space using random mutagenesis. So the first strategy involves making better libraries for selection experiments. So what we mostly mean by this are libraries with a higher fraction of active sequences. So the goal here was to make a library in which most or all of the sequences had the potential to form the secondary structure of Aurora, and were also consistent with the sequence requirements of Aurora. So to design this library, we used information from a data set of about 100,000 variants of Aurora that we identified in a previous selection experiment. So the idea here is to find a way to encode the variation from these many sequences into a small number of oligonucleotides. And we also wanted our strategy to be consistent with a standard DNA synthesizer because these kinds of instruments are widely available. So to generate this library, we classified the positions in Aurora into 3 different categories. The first category I'm calling constant. These are shown in green, shown in blue. So these correspond to positions that were highly conserved in our data set of variants, so we didn't mutagenize them at all. The second category I'm showing in purple. These are positions that occur in unpaired parts of the secondary structure and exhibit some variation. So here's one example here. Sometimes this position is A, and sometimes it's G in our data set. So we encode this with R, which is 50% A, 50% G. That's something you can do using a standard DNA synthesizer. The final category of positions are, for the most part, positions that form base pairs in the secondary structure. We're showing these in green. And these were encoded using pairs of degenerate positions. And we chose these pairs in such a way that we both maximize the representation of different variants of the base pair, while at the same time minimizing the probability of breaking the base pair. So there are many ways this kind of encoding can occur. It's kind of an interesting problem. We wrote a small paper about this a few years ago, and I can also talk to you about this afterwards if you're interested. All right, so using this sort of approach, we generated a library that encoded 17 million sequences, and of these, 5 million were consistent with the sequence requirements of Aurora. And this required 8 different oligonucleotides that we mixed. So the next step was to identify the catalytic molecules in the library, and we did this using a method called single-step selection. So the first step is to mix the library with the 4-MUP substrate. The active molecules will transfer the phosphate group from 4-MUP to their own 5' hydroxyl. That's the reaction catalyzed by Aurora. We then perform a ligation reaction. And the important point here is that this ligation reaction requires a 5' phosphate. So it means that only the reactive library members get modified. These can then be purified by Page because they're now longer than the unreacted library members. Finally, they're cut from the gel, amplified by PCR, and characterized by high throughput sequencing. So this graph at the top shows the result of this single-step selection experiment. So on the x-axis, I'm showing CPM, counts per million. So this is the number of reads of the sequence times 1 million divided by the total number of reads. So it's a measure of the frequency of the sequence. On the y-axis, I'm showing the number of unique sequences with the corresponding CPM value. These red columns indicate the distribution of CPM values in the starting library. So this is before any selection. And the gray columns show the same distribution after 1 round of selection. So if this library didn't contain any active sequences, the red and the gray distributions would be identical, but that's not what we see. We see a lot of shifted sequences, so that indicates that the secondary structure library contains many catalytic molecules. We also did this using a control library in which mutations were generated by a random mutagenesis. This is kind of the standard approach in the field. And you can see that in this case, there are very few catalytic sequences. So by comparing these two graphs, we can say in a qualitative way that the secondary structure library contains many more active variants than the randomly mutagenized one. One other point I want to make here is that both of these libraries encode an internal control. This is Aurora 2, our best previously identified variant of Aurora. And you can see that this is shifted in both cases. So this tells us that from a technical point of view, both of these selections worked. So we wanted to compare these two libraries in a more quantitative way. So to do this, we chose a number of sequences from both of these libraries over a range of CPM values, and we tested them for catalytic activity using the ligation assay. So this graph on the left shows the relationship between CPM and activity. So what you can see is below CPM values of about 10 or so, there's not really a correlation between CPM and activity. So this tells us that these low values are noise. However, above values of about 10 or so, we see this nice correlation between CPM and activity. So it tells us we can use CPM as a readout for the catalytic activity of library members. One other point I want to make is points from the secondary structure library shown in blue and the randomly mutagenized library shown in green more or less fall on the same line. So it tells us we can compare CPM values from different libraries, at least in this case. On the right, I'm showing a comparison of the number of unique sequences in both of these libraries using different CPM cutoffs. So if we use a cutoff of 10, which seems to be reasonable, we conclude that the secondary structure library contains 40 times more active sequences than the randomly mutagenized one. So for us, this was encouraging and exciting. Another sort of advantage of the secondary structure library is that it explores more diverse parts of sequence space. So in this case, we're looking at sequences with CPM values of 10 or higher, and we're looking at pairwise distances. And what we can see is the sequences from the secondary structure library are shifted relative to those in the randomly mutagenized library. So the take-home message here is that the secondary structure library helps us to explore more diverse parts of sequence space. The secondary structure library also contained improved variants. So on the left, we're showing these variants plotted as a function of mutational distance from Aurora 2, our previous best sequence. On the right, I'm showing a head-to-head comparison of one of these variants called H1 with our previous best variant, Aurora 2, and this was again done using the ligation assay. Okay, so to summarize this first part of the talk, secondary structure libraries appear to offer several different advantages compared to randomly mutagenized ones. First, they contain more active variants. Second, they contain more diverse active variants. And as an added bonus, we found in this case that our secondary structure library contained improved variants. All right, so I'm now going to move on to the second topic of today's talk. And the idea here is to combine the power of artificial evolution with the power of machine learning to try to explore sequence space more efficiently for active molecules. So the goal here is to make a model in which the input would be a new sequence, a sequence that the model has never seen before, and the output is the correct activity. So originally we tried to make such models ourselves, and it didn't really work out. I then talked to another group leader at my institute named Tomasz Pluskal, who's an expert in machine learning. He suggested that we try this approach, and he even agreed to help us. So in thinking about whether this is even possible at all, it's important to consider the rules that relate sequence to function. How complex are these rules? Well, this depends on something called epistasis. This can be thought of as a measure of how variable the effects of a mutation are when you test the mutation in different backgrounds. So imagine, for instance, an A to C mutation at some position in Aurora. If epistasis is low, the effect of this mutation will be more or less the same in every background you look at. And in these cases, modeling is quite easy. On the other hand, if epistasis is high, the effect of this mutation will be more or less different in every background you look at. And in this case, modeling is difficult or even impossible. So we wanted to know how much epistasis there is in Aurora. So To address this, we used the following workflow. We first picked a reference sequence. Again, it was Aurora 2, our best previous variant. And we determined the effect of every possible mutation in the library in the Aurora 2 background. We then looked at the effect of each of these mutations in every other background in the library. And finally, we compared the effects of each mutation in Aurora 2 with the effect in every other background. So here is the disturbing result of this analysis. So what I'm showing on the x-axis is each of the possible mutations that can occur in Aurora 2 in the context of this library. And on the y-axis, I'm showing the fold difference in mutational effect relative to Aurora 2. So if there was no epistasis here, all of these values would be 1, and modeling would be quite easy. But that's not what we see. At every single position, we see this large range. So the effects of mutations generally can change by about 1,000-fold in different backgrounds. So that's pretty insane, and it tells us that the rules linking sequence to function in this case are quite complex. But despite this, we were able to develop models by machine learning that do a pretty good job of predicting CPM values of library members. So on the left, I'm showing the workflow. So the starting point here
Günther, question. I can walk that.
Thanks. Thank you for this very interesting talk. So I have a, maybe that's a little bit provocative question, but it also bothers our own work. So you are doing selects to identify deoxyribozymes that catalyze a specific function. And then you're using machine learning to optimize the selected sequences. So to which extent do you think that the machine learning process would be much better to optimize our selection process? So because we are limiting ourselves in a way that we apply a selection scheme that give us a handle on either, and then we take it from there with machine learning. But wouldn't it be better that we would apply machine learning to the selection process itself? Because if you compare our ability to generate optimals, for instance, in our world for small molecules, we are much far behind nature. They can do it much better. So I don't know, what are your thoughts on this?
Okay, well, I agree with you. And my feeling is we would need a huge amount of information to do this. And I don't know if that's true, but that's my general sense. So, you know, in this case, well, what can we do? We can fairly easily generate these gigantic data sets containing millions of variants of one particular motif. So that seemed to be a reasonable place to start. But let's say we were trying to make this more general and we wanted to train machine learning so it could maybe tell us, here's the best sort of library to use for this particular experiment, or maybe even better, here are some actual sequences that you can use right off the bat. Well, would that be possible? Perhaps, but I would imagine you would need an enormous data set of variants. Okay, but we haven't tried this yet. So maybe we already have enough. There are huge data sets. I mean, we were talking about this the other day, and it's a really interesting question. But at least so far, we thought this kind of approach would be simpler. And well, we didn't even know if it would work. So we start here and then maybe move to something more complex like you're suggesting. But I agree with you that this would be fantastic, especially considering how time-consuming and difficult selections are. And in our lab, well, you know, this is a never-ending story because a student gets good at selection and then what happens? They graduate. And then I have another student who's smart, ambitious, but still needs some time to learn. So absolutely, if we could just sit on the computer, this would be a dream. And well, maybe someday it will happen. Maybe before I die, it will happen. Not to be grim here, but that would be super cool. So.
Thank you. Okay. Awesome. Well, thank you so much for the talk. Really interesting stuff. And apologies if this is like an ignorant question, but I was curious if for the machine learning model that you trained on Aurora, if when you tested it on the other DNA zymes, did you retrain it on that new data set, or did you? So then I guess a follow-up question is then, did you ever try sort of seeing if you train the model on Aurora, is it still able to predict the CPM values for other like DNA zymes without being trained on that, just completely unseen?
Yeah, so this is something that we've talked about in the group, and basically the idea would be to train a model using all of the data sets we have, and maybe even going beyond that and training it with other data sets that are available. We haven't yet done any of that, and I would imagine it's going to be a much more difficult problem, but well, we haven't tried it, and it's like something quite interesting, you know? So it's a great question.
Thank you.
Very good talk. I was wondering, for the candidate that performed better than the original Aurora, do you know the mechanism why it performed better? Like which type of nucleotide that makes it perform, that has better catalytic activity?
Well, I would say yes and no. I mean, we know the sequence, but we have no idea what these mutations are doing. And I think something that would help us a lot would be to get a structure. There aren't so many structures of deoxyribozymes, and well, we've been collaborating with people for a number of years on this, and we're making baby steps, but still don't have a structure. But once we do, I think that's going to be really informative, and I'm also going to be very, very happy.
Thank you very much.
Very nice talk. I have a question about when you training the model with Aurora. Have you limited the size of the structure? How long usually you use, except like, oh,
So sorry, I didn't
the
understand.
the sequence size? Like
Oh,
how
yeah.
long you
Okay,
usually
so
use?
the sequence of Aurora is 47 nucleotides, but
Okay.
in this particular library, I think only 24 positions are mutagenized. So that's basically what's being fed into the model, and no other information, just the sequence of each variant, and then we see how well can it predict the CPM.
Okay. Is it possible you can expand more longer, enzyme will be work for the model?
So you mean, can we apply it to other deoxyribozymes?
Yeah, or longer one.
Oh,
It be same.
well, I mean, I would say in principle, yes, but we don't yet know until we've
Yeah.
tried.
Maybe you can try.
Thank you for the talk, Edward. I guess I don't understand something. So you're calculating CPM based on the sequences,
Yeah,
and you're using that information to feed in for the prediction.
yeah.
And you're not taking the low CPM values, because in the aptamer field, sometimes it's the low frequency
Yeah.
sequence results that you end up with the real aptamers. You haven't tested those actually, all those sequences in terms of catalytic activity, have you?
No. So basically, so we have CPM values for, I think, like 1 million sequences or so. And it's not really feasible to test them all. So we simply tested some, and we're assuming that these were representative sequences. So that's basically it. So if there wasn't a correlation here, like for example, in this part of the range, there's not, we say, forget it. But one other thing to say is, when we trained the model, we didn't say, forget it. So in other words, we randomly chose sequences, and that's what the machine learning used. But this was kind of discussed at length in the lab. Is this the right approach? Should we be omitting these sequences because the difference between this and this is really not significant? Okay, but the other side of it is exactly what you said. The dead sequences are also providing information. And another thing that some of the machine learning people said is, sometimes this can be a problem if you have an unbalanced data set. And in this case, there are many, many more sequences that have low activity than have high activity. And that seems to be kind of a general property with functional nucleic acids, that as you get better and better, the number of variants above this cutoff for activity goes way down. But I think there are many, many aspects of this that we can play with and we don't really know. And that's what I'm hoping we'll address in the next few years.
Okay, if there's no other questions, I have one.
Oh, yeah.
So, if you think you go ahead one slide, you had the secondary structure diagram
Yeah.
somewhere there.
Like
So yeah, it looks very plausible. I guess if what you're doing is varying certain what are base pairs here, or some aren't in base pairs, with the better what the structure is, help you either limit the number of sequences you're predicting or maybe get better predictions?
Yeah, it's possible. I mean, right now have the secondary structure model that's we made by comparative sequence analysis, and
Mhm.
we tested these proposed base pairs using mutant cycles, but we don't have any NMR
So-
data for this. We don't
Yeah.
have any crystallography data.
Yeah.
We even talked about cryo-EM, and I think it's a bit too small. but,
Yeah, probably.
I don't know,
But-
but yeah,
If you took a deoxyribozyme or even an aptamer with a known structure,
yeah.
would that make the predictions easier or
Yeah,
better
I-
or?
I would think that the answer is yes, and that could actually be an interesting starting point. And
Yeah.
maybe a variant of this approach would be, okay, it's really hard to get this structure, but if we take something that already has a structure and then we do a selection experiment, then we
Mhm.
can also generate this huge data set, and we can do that fairly easily. So
But-
that could be quite interesting to combine those approaches and maybe try to see how much information the structure is giving us.
So what sort of data set do you need? You need an aptamer selection data set,
Yeah,
knowing the structure,
yeah.
but do you need to know the binding data for all the different variants? Is that
Well,
the idea?
not for predicting activity, but that's another interesting question. And we recently did an experiment in which we basically randomized part of this molecule and did a selection for variants that could use new substrates.
Hmm.
And so we basically found that point mutations at these 4 positions can change the substrate specificity. We actually used these data sets also for machine learning, and it worked to some extent, not as well as in this case where we were just looking at activity, but
Okay.
that's also a really interesting question. And again,
Yeah, we should
yeah,
talk afterwards then.
absolutely,
It's really
absolutely.
cool.
Yeah.